Thank you everybody for coming here and thank you Living Science for inviting me to give this lecture. So Living Science is doing a great job in promoting multidisciplinary science and research. And today I'm going to talk about microfluidics, a field which is truly multidisciplinary. Microfluidics lies at the interface of engineering, physics, biology and chemistry. So we can go into depths of physics, engineering, biology and chemistry if we want to talk of microfluidics, but we have a general audience today from physics, engineering and biology. So I'll talk of physics and applications because you can't talk of applications and physics separately. In particular, if you want to see how this field is, you want to understand the power of this field, you also need to understand the physics based on which you can find new applications. But overall goal of this talk is on developing applications for biotechnology. And this is more of a teaching lecture, a teaching presentation. So I have taken figures and videos from different sources. So I might not attribute or refer to those actual sources from where I have taken, but that's because this presentation is only for teaching purpose. And because we are talking of applications, it's important to talk of the companies which have been spun off from this field. So that make sure that I would like to clarify that if I mention a name of a company, it's by no means that I am endorsing that company. I am telling that I would name those companies because as an engineer we should make sure that whatever we discover in the lab, we should actually translate that into an actual marketable product. So that is why I would name those companies. So let's look at how we handle liquids in a conventional biotech or chemistry lab. So what we have and what you would be familiar with is these pipetting techniques where you can do at max 10 pipetting per minute, and the volumes that you handle are about 1 milliliter or about hundreds of microliters. So what you see over here is a 96 well plate where you can do 96 let's say parallel assays. But if you are lucky, you can get a robot like this shown in this video which can do this job for you. But still at best you can get about 100 pipettings or 100 different operations per minute, and that's not a large rate at which you can do these operations. So being an engineer, so this process actually seems to me like doing calculations with hand, and when you do these hand calculations and you want to scale up, what would you do? And what engineers did was that they invented the integrated circuit. So the question is that can we have an integrated circuit for chemistry and biology where you can automate and integrate these processes, let's say on a single chip, so that you don't have to do these processes manually, and you can do a lot of processes in parallel and at very fast rates. So that is the motivation behind this field of microfluidics. So what we do is that we develop an analog of integrated circuits for handling fluids. So just like you have a silicon chip in microelectronics, we also have a microfluidic chip in this field. So what is a chip? So in a microfluidic chip, you generally have a large network of channels through which you can flow fluids, do processes such as mixing, and then have some reactions, do some thermal cycling and analyze the products. So essentially whatever processes that you do in a lab, you can do it on a small chip which is size of few centimeters by few centimeters. And typical channel size is about 10 to 100 micrometers. So that's about 100 micrometers is about the diameter of a human hair. Okay, so the fabrication of these chips are not so difficult because you can borrow techniques from microelectronics. So in microelectronics you have feature sizes in nanometers. Here we have feature sizes in about tens of microns. And you can do nanofluidics, but the choice of about 10 microns is that size diameter of a channel is because a typical particle size is 5 microns. So in Delhi you would hear about PM10, PM5. Okay, so 5 micron is a typical particle size of a dust particle. So if you have about 10 micron size channel, then you need not worry about dust. So you can develop these microfluidic chips in a large variety of substrates, in glass, in silicon, in plastics, in elastomers, and recently people started making these chips on a paper itself. And the advantages of this field come when you integrate large number of lab processes onto a single chip. And to give a demonstration, so let's look at this chip which was developed back in 1998 to integrate processes for DNA analysis. So just as I showed you in the second slide, that you can do pipetting, you can do mixing, and then you take those reaction products and go for further analysis. This chip, which is of a size of few centimeters by few centimeters, can do all those processes in an integrated fashion. So if you look at this chip, so you load a sample, then it will measure the volume of that. It will precisely take that particular amount of volume that it needs. Then it will mix some reactants, and there's a region where you can have some thermal reaction, such as restriction, enzyme digestion, or PCR, thermocycling for PCR. And once you have got the reaction products, you can do some kind of separation. In this case, it is gel electrophoresis to detect the reaction products. So imagine if you can do all on a single chip. Okay, and the advantage of doing this is that rather than using large volumes of reagents, you can use tiny amounts of reagents, and that reduces the cost. So as I told you earlier, the smallest volume that you can handle, let's say manually, is about 100 microliters. But what you can do in these micron size channel is about picoliters to nanoliters. So that's a huge reduction in cost of the reagents that we are using. Now the difference between microfluidics and microelectronics is that when you reduce the size of electronics, unless you go to really small nanoscale, the physics does not change that much. So it's more about just integrating those transistors on a chip. But when you reduce the size of your fluidic channels, the physics changes drastically. And based on this new physics, we can develop new applications. So that's why if we need to understand the applications, we should also know a little bit of the physics behind flow in these microchannels. So the flow at microscales is very different from flow which you see around yourself. Now to begin with, let's look at this video where you are mixing milk with tea, and you know that, so this video is in slow motion, but you know that when you pour milk in tea, you see that suddenly the mixing takes place, and there is some chaotic mixing which you can see, and that's because fluid flow in macro scale is characterized by turbulence. So you see turbulence all around yourself. But what happens is that if you go to micro level, the flow physics changes completely. So at micro scales, the inertial forces which are responsible for turbulence are much lower than the viscous forces. Okay? And the ratio of these inertial forces to the viscous forces can be characterized by a dimensionless number which is called the Reynolds number. Okay, so at low Reynolds number, you have no turbulence and the flow is linear. So there is no non-linearity, and that means that you can handle fluids very precisely, and there is no uncertainty in how the flow will behave. Okay, so here is a video on the bottom. You can see flow visualization of this low Reynolds number flow in a microchannel. So the thickness of this channel is about 90 microns. So I've seeded those the flow with fluorescent particles, and you can track those fluorescent particles, and you can see that the flow is very well behaved. It is laminar. Now, how can we use this well-behaved flow to our advantage? So, the first example which is a most simple example in microfluidics, is an example of a T sensor. So, before going into the working principle of T sensor, let's look at this beautiful image over here. So, this image is of the confluence of Indus River with Zanskar in Ladakh region. So, the Indus River comes from the left hand side, that is in green color, and the pristine blue color is of the Zanskar River. And you can see that at the confluence, you do not see mixing suddenly. So, there is a gradient that is established. So, now let us shrink this gradient and shrink this whole system to a chip, okay, where the thickness of the chip is about 100 microns, and again you will see a similar phenomena, but instead of having turbulent mixing, you will have only diffusive mixing, which is really slow. So, what we have over here is that we have two parallel streams of flow, and let us say one stream has some chemical species mixed in it. So, those species would diffuse to the parallel stream which has lower concentration. Okay. So, due to a concentration gradient, we will have interdiffusion zone. Now, this interdiffusion zone is actually very small compared with the thickness of the channel. To understand the physics, for a physicist, they are familiar with this diffusion time scale. So, if you have a length scale W, in this case the width of the channel, and you have a particular diffusivity, the time taken by a diffusing species to fill this width would scale as the squared of the width of the channel divided by the diffusivity. Now, during the time this diffusion is taking place, the solute will also convect along the length of this microfluidic channel. So, the distance covered would be the speed of the flow times the diffusion time. So, if you take the ratio of these length and the width scales, you get a number called Peckley number. So, typically this Peckley number is order 100. That means you need to travel 100, let us say, millimeters to get a diffusion zone of 1 millimeter. Okay. So, that means in general you will have a very narrow interdiffusion zone, and recall that the flow is laminar. There is no chaos or chaotic processes going on, there is no uncertainty. So, everything happens very precisely. Okay. So, here is the advantage of microfluidics. So, in this picture on the right hand side, what we are doing is that there are two streams, one which is laden with a dye which binds with calcium. So, in the other stream we have calcium chloride. So, initially these two streams are not mixed. Now, when these two streams mix, there is an interdiffusion zone, as you already know now, and in this thin region the dye binds with calcium and gives fluorescence, which you can detect over here. Okay. So, this is something like a flow reactor. So, you can understand reactions. So, if you visualize this reaction, the length along the channel is just like a time coordinate. So, as the solute moves from the inlet to the outlet, it is just like how it will behave when this reaction takes place over time. Okay. So, you can visualize, and people have used it to measure properties of chemical species such as diffusivity and reactivity and the reaction constants, which are really important to characterize certain kind of many kind of reactions. But I am going to focus at a particular application, and that is on doing a competitive immunoassay, because the overall aim of today's talk is on how we can use this interesting physics at microscales for doing biotech related experiments. So, we have we can do a competitive immunoassay, because in immunoassay what happens is that you have an antigen and you have an antibody and they bind with each other. So, now you can understand that you have two parallel streams. One, you can fill it with an antibody, and the other stream is, let's say, laden with a labeled antigen. That means it is fluorescent. Okay? And you should also, and as is obvious that if you have a larger molecule, it's going to diffuse less. So diffusivity depends on the size of the molecule. Okay? So here are two parallel streams coming together, one with antibody, and let's say one has only a labeled antigen which gives some fluorescence. So when this reaction takes place in this narrow diffusion zone, the interdiffusion zone, this labeled antigen, it binds with a very heavy antibody. As a result, its diffusivity decreases. So, okay, so now it's bound to a big molecule, so its diffusivity decreases. So what happens is that its ability to diffuse into the other stream goes down. So what you end up is, if you look at this figure, the fluorescence intensity in the span-wise direction, you get a hump in the fluorescence signal. Okay? So now what you do is we do a competitive immunoassay. So if there is a sample antigen present in that stream, it will compete with reaction with this labeled antigen, which you will add from your own side. Okay? So this hump in this fluorescent intensity would go down if you have a sample antigen which is not fluorescent. Okay, so it is competing for the same binding sites on antibody. So looking at this height of this deviation in the fluorescence signal, you can actually quantify the concentration of unlabeled antigen which you want to detect. So people have done this. So on the figure in the right hand side, you have blood which has particular antigen, and you add a labeled antigen from your side. You make it flow parallelly with an antibody which is not tagged with any fluorescent species. Now if you observe the fluorescence intensity, you will see that you will have a local maxima in the fluorescence signal, and looking at the height of this, looking at the maximum value of this signal in the middle of the channel, you can actually quantify what is the concentration of the unlabeled antigen. So with a very simple device, a passive device, you can do a competitive immunoassay. Okay? And keep in mind that for an immunoassay you need a lot of reagents, and now we are using only nanoliters of reagents. So proportionally you can reduce the cost of your analysis. Okay, so we've looked at one application where we see that this works on the principle of differential diffusion rates of small and large species. So we'll look at the next application. So based on the same principle, we have a device called HFilter. Okay, HFilter is again a passive device which can purify a sample without having any membrane. So you can filter without a membrane. So the idea is that, let's say in this figure you have large particles and a mixture of large and small particles. Okay, so they can be two chemical species, one which has high diffusivity and low diffusivity, and you want to extract only that species which has higher diffusivity, then this device would work. So now you have, in this device, you flow two parallel streams again, one with a mixture of these large particles with low diffusivity and small particles with high diffusivity, and flow it parallel to a dilute stream or a stream which has none of these particles. So as these particles flow along the channel, intuitively you would understand that smaller particles are going to diffuse faster, and if you choose your Peckley number, which is the ratio of the length of the channel and the width over which these solutes can diffuse carefully, then you can make sure that the stream that is going out from the top will have proportionally large amounts of only the higher diffusing species. Okay, so and you'll have a waste stream where you'll have which goes to the bottom, which will have both high diffusivity and low diffusivity species. So essentially you can purify or you can reduce the amount of high diffusivity species among the low diffusivity species. And there are plenty of applications for this, but I am going to focus on one application, and it's an interesting application. It partly behaves on this random motion which we call as diffusion, but it's not exactly based only on diffusion. So we look at this application. So this is again a use of HFilter to separate motile sperm from a non-motile sperm. So sperm, if you have a mixture of sperm in which you have some healthy sperms and let's say dead sperms, and you want to extract only healthy sperm. So it has a lot of applications. For example, in IVF treatment, you would want to look for a healthy sperm, okay? Or let's say you can use these kind of devices for detection of infertility. So you can make this passive device. So again, it has an kind of H-shaped channel. From the top channel, you flow the mixture of motile and non-motile sperms, and then you have a dilute stream or a stream with no sperms flowing parallelly. So what happens is that a dead sperm or a non-motile sperm would diffuse just due to molecular diffusion, okay, just by random kicks from the fluid particles, okay, and it's a large size particle, so it's not going to diffuse. But the motile sperms or the healthy sperms, they are going to they will not diffuse, they are going to actively move, but in all random directions. So as soon as they enter the channel, they are going to completely fill the channel, okay. So if when you completely fill the channel, you can extract only those that part of fluid which has only the motile sperm, and the non-motile sperm which does not diffuse, you can collect it from the top stream, okay. So here's a video of this process. So you can see that the dead sperms would continue moving because they are diffusing at a very slow rate. So they would just be carried by the flow, whereas the motile sperms would fill the channel, and you can collect them from the bottom channel. Okay, so people have commercialized this kind of technology. So there's one company called Koik Biotechnology in Turkey, which have commercialized a technology which works on a similar principle, but not exactly on HFilter, but it works on the differential diffusion rates of motile and non-motile sperms. Okay, so it's a passive device. You put in the sample and collect a sample which has a larger fraction of healthy sperms. Okay, so imagine that how much reduction in manual labor and time can take place for a person who is doing IVF treatment. Okay, so this is these are examples of microfluidic devices which use low Reynolds number flows. Okay, and low Reynolds number flows are easy to understand because at low Reynolds number, where viscous forces are dominant, the flow is linear. So it's always easier to understand linear processes, and but that is relatively not so interesting because you know how the behavior is going to take place if the process is linear. So to introduce non-linearity, we can have two fluids now, and two fluids which are immiscible. Okay, so this field is called droplet microfluidics, and it has a lot of applications. So let's see what we can how we can generate these droplets. So consider a channel shown on the top over here. We have a T-shaped channel. Okay, so let's say from the bottom channel you are pushing in water, and from the side channel you are pushing in oil. Okay, so if your channel or the microfluidic chip has walls which are hydrophobic, then oil will wet the walls and you will form water droplets. So in this case we have an additional force which is the interfacial tension, okay, with the coupling of interfacial tension, the competition between interfacial tension and viscous stresses, you form these droplets. But the difference is that you can do this in macro scale, but the difference is that you can do it very precisely on micro scales. So what you end up is, as shown in this video, you get droplets of uniform shapes. So same shapes for every droplet. So there's no polydispersity. Okay, so these droplets are monodispersed. You can have monodispersity of the order of 0.5%. So, and the rate at which you can generate these droplets is really high, about 1 to 10 kilohertz. That means 1000 to 10,000 droplets per second. Okay, so this is the rate, and typical size of the drops is about the diameter. Let's say if the droplet is spherical, is about 100 micrometers. So if you calculate, the volume is about a nanoliter. Okay, now how can we use these drops to our advantage? Okay, so as you can see over here, you have oil surrounding a water drop. Okay, so if there is a chemical, there are some chemical species in these water drops, they are not going to diffuse into the oil stream. Okay, so what we can do is that we can treat these drops as individual reaction chambers. So if we do some reactions in these drops, you can do thousands of and millions of reactions in parallel. So it's an analog of your 96 well plate on a microfluidic chip. Okay, so because you can generate thousands of drops in a second, you can initiate trials for thousands of reactions in parallel. And again, keep in mind that we are using nanoliters of the reagent, so it brings down the cost. So one example of this is in protein crystallization. So those working on protein crystallization would know that, you know, to get a crystal for X-ray crystallography, you need to get a good crystal, and to get a good crystal is a very daunting task. You need to screen a lot of conditions in the phase diagram, the crystallization phase diagram, to get the optimal conditions at which the protein will crystallize. And typically this phase diagram is multi-dimensional and very complicated. So what you need to do is that you need to do combinatorial chemistry experiments. That means, let's say if you have two reagents and you need to try all possible combinations to find the optimal chemistry at which the protein is going to crystallize. Okay, so what we can do is that in this schematic that I'm showing over here, so let's say you have two reagents A and B flowing, let's say these reagents are aqueous based, and you have a carrier fluid which is let's say an oil. So as soon as the droplet forms, a reaction is initiated, or these reagents mix, and you can, as you know, that you can do initiate about 1000 reactions in parallel. So by just controlling the flow rates, the relative flow rates, you can change the concentration of these reagents in individual drops. Okay, so you can actually initiate thousands of crystallization trials in a few minutes. Okay, and then you can see that in this picture, people have found optimal conditions for doing protein crystallization. So imagine doing these 1000 reactions manually will take you a lot of time, but you can initiate them all in actually 1 second, provided you control the flow rates with the computer. You can initiate these crystallization trials in a very short time. So that is one application, and you can imagine there would be many other applications, because for getting an optimal reaction, you would need to look at the right conditions in the parameter space. Now let's look at another application of droplet microfluidics, and that has to deal with PCR, and you do it day to day, but those in physics and engineering would not know about it. So here's a brief introduction of PCR. So this technique is used to amplify DNA strands. Okay, so if you have a single double-stranded DNA fragment and you want a large sample of same DNA fragment, you can amplify that using this process. So in PCR, you start with a double-stranded DNA, then you heat that mixture and this process is called denaturation, and you denature the sample and the two fragments separate from each other. Now your reaction mixture has some primers which bind to these, which are small fragments of DNA which are complementary to these DNA strands, and they bind at the ends of these DNA strands which are now separated from each other. And then if you have the building blocks of DNA, A, T, G, C, the nucleotides, and an enzyme which is called DNA polymerase, then at a particular temperature, if you cycle the temperature, it will build the DNA fragment. So after one complete cycle, you will now start with 2 fragments, you will have 4 fragments. If you repeat this process, you can understand that, visualize that this process will exponentially grow, and you will have 2 to power n strands after a particular number of cycles. Okay. So how do you monitor this? So there is a process called real-time PCR, in which you add a kind of dye which will give fluorescence proportional to the number of new fragments that you have generated. So just looking at the fluorescence, you can quantify how much how many new fragments which you have generated. So you get a signal like this shown over here, fluorescence versus the PCR cycles. So this diagram you can use to quantify how many fragments you initially started with. So you can define a threshold at which, let's say you have 10 cycles, in 10 cycles you cross this threshold, you can estimate how many fragments I had initially. But there are some issues with this traditional PCR, and that is that as you understand, the fragments increase exponentially with the number of cycles. So it's a highly nonlinear process, so small uncertainties are going to get amplified. Okay, and you need a calibration experiment. So if you have some targets, you need to put a known, in a separate experiment, you need to put a known number of targets to see at how many cycles you will get that threshold. Okay, so to avoid that, there's a process called another technique called digital PCR. Okay, and this was known before the, droplet microfluidics came into the field, but there was people didn't knew how to actually do digital PCR. There are other ways to do digital PCR also, but I'm going to talk only about the droplet microfluidics approach to do digital PCR. So let's see what is digital PCR. So digital PCR, as you understand from digital, is that you have on or off, 1 or 0. So let's say you have a few fragments of DNA, as I have shown over here, and then you take a lot of test tubes and dilute this initial sample in a way that every test tube has either 1 fragment of DNA or no fragment. Okay, so this is the most basic way of doing, most simple way of doing digital PCR. Okay, so let's say somehow we can do this kind of dilution where these test tubes have either 1 strand or no strand. Okay, now in parallel, if we start doing PCR, okay, only those test tubes will show fluorescence signal which actually had a single strand. Now you just need to count those test tubes which had single strand, and you exactly know how many strands we actually started with. Okay, so this is called digital PCR, and in general you can have actually 2 strands or 3 strands also, but you know, by Poisson statistics you can take care of that kind of situations. But the overall process remains the same, that you dilute and just see, do a large number of PCR reactions in parallel, and now you understand and you would guess why my digital PCR can be done in droplet microfluidics. So here's the process: you generate droplets, okay, and you dilute your initial sample such that every droplet has 1 DNA strand or no DNA strand. Okay, so let's say you have a microfluidic chip in which you can generate these droplets, and you can do millions of these droplets. You collect that sample. Now in your existing thermocycler in your lab, you can collect these droplets in 96 well plate. So every vial in the 96 well plate can have large number of droplets, that doesn't matter. Okay, and these are water droplets in oil phase. So you thermal cycle to the end point, okay, and then you make a chip, a microfluidic chip, in which you flow these drops again, okay, and do a process which resembles flow cytometry. You quantify the fluorescence in individual droplets. Okay, so in that way you can do this digital PCR with your existing setup in the lab with some additional microfluidic chips. Now you would say, why is this digital PCR needed? So one application of this is that, let's say you want to detect a mutation in a DNA fragment among which is mixed with a DNA fragment, a wild type DNA fragment, which is in relatively large quantity. So let's say if you have a one fragment which has a particular mutation among 10,000 DNA which don't have that mutation. So if you do your traditional PCR, you would understand that, or you would realize that the wild type DNA would overwhelm the signal from the mutated target. So in this way you can actually look at these kind of applications where you are actually targeting a very rare allele. Okay. Similarly, for copy number variation, you can use this digital PCR, and this technology has been commercialized by Bio-Rad. In fact, this paper was published by Bio-Rad back in 2011 in Analytical Chemistry.